Oh, it's my great pleasure this afternoon to introduce Dr. Nicholas Wu to give the last real seminar for the year. And he's going to be talking about chytrid fungus and how it disrupts the skin physiology of frogs. Before I pass it over to him, though, I'd just like to give a quick bit of background about him. So he's originally from across the pond in New Zealand, where he completed his undergraduate before he then moved across to Australia in 2015 to the University of Queensland to complete his honours and PhD, where I had the great pleasure of sharing an office with him for a couple of years. After completing his PhD in 2019, he then moved down to the University of Sydney to work with Frank Seabarker, looking at the effect of PFAS on fish physiology. And then once that wrapped up in 2022, he's now started his second postdoc working at the University of Western Sydney, where he's now looking into the sort of physiological underpinning of what makes bats susceptible to white nose syndrome. But without further ado, I'll pass it over to him to present his talk for the day.
Thanks, Cameron, for the introduction. And thank you as well for Hannah and Chava for inviting me to give this talk and for everyone here to listening to it as well. I do also want to acknowledge the Darug people, the Aboriginal owners of the land where I'm speaking to you from, and pay my respects to the elders past, present, and emerging. So, amphibians are a really unique group of species, and I have a really personal attachment to them as well. They're a really nice, diverse group of animals to work with, but unfortunately, a lot of them are in decline. In fact, over 6,400 of the described species that are known, 43% of them are listed as threatened. And so that's roughly around 2,500 species that have declined. And a lot of them actually have been declining in the more recent decade as well. There's this nice figure back in 2004 that shows what are the causes of these declines, and they can be grouped into three main factors. One is habitat destruction, second is overexploitation, and the third category is enigmatic. And so you can see on this map here, there's a lot of areas that have been caused by habitat destruction in the green here. But there's also sections that have been, areas that have been, amphibians that have been overexploited for medicinal purposes, pet trade, and for other consumptions by humans as well, mainly around Southeast Asia and also in Southern America as well. But if you have a look here, there's a lot of red areas in the Americas and also in Eastern Australia as well that are labeled as enigmatic declines. And these are associated with declines that are not directly impacted by humans. And so I wanted to say before that, okay, there's a lot of declines happening in those areas. But then this year, there's this new paper that came out that had revisited these risks associated with these declines and included the effects of climate change as well. And so there was an update on the amphibian risk. And now it seems that climate change is the major contributing factor to amphibian declines in the recent times. But if you have a look here, enigmatic declines, or what they call diseases here, are still prevalent across the globe as well. And so what I wanted to say originally is, what are the causes of these enigmatic declines? Because they not only occur worldwide, but they occur in areas that are not really occupied much by humans. So they're usually pristine mountain regions. So for example, the cloud forest of Central and South America and the montane rainforest of Queensland and New South Wales. And there are a number of potential causes that have been shown to directly cause declines in amphibian communities and individual species as well. So they include introduced species that are both in competition with the native species there, or as introduced predators as well. There's also pollutants that include plastic pollutants, agricultural pollutants, pharmacological pollutants as well, that have been known to impact a lot of amphibians. Disease is a really prevalent factor, especially in the 21st century, and of course, climate change as well. So not only global warming, but an increase in UV radiation as well from the depletion of ozone layers. But what I wanted to focus here is on the disease aspect, because the occurrence of infectious diseases have been rising a lot, not only in animals, but in plants as well, and of course in humans. And so you can see on this graph here, there have been a number of extinction events directly caused by fungal pathogens that has been on the rise recently, and especially in animal cases as well. And so one of the major pathogens that has been affecting amphibians is known as Chytridiomycosis. It's a lethal fungal disease caused by the pathogen Batrachochytrium dendrobatidis. We'll call it BD for short here. But just to note as well, there is another sister species that was discovered in 2013 called B cells, so Batrachochytrium salamandrians, that also affects salamanders as well. So they have different hosts, but induce the same kind of pathological disruptions as well with them. But for this purpose, I'll be focusing on BD here, because it's where I did most of my study on. So BD is globally widespread, and it has affected over 500 species of amphibians worldwide. And because of that, it is known to be responsible for the largest known decrease in vertebrate biodiversity caused by one single disease. So it's found everywhere where amphibians are. So it's truly a global event. What is this fungus, and what does it actually do to amphibians? Well, it has a really simple lifestyle. It only has 2 stages. It starts off as a motile zoospore. So it's free-flowing in the water, and it's attracted to the keratin on different animals, and amphibians have keratin on their skin as well, so it's attracted to that. They'll swim towards the keratin on the skin, embed themselves in there, and start growing within the top layer of the skin, the epidermal layer. And from that, they'll transform into a zoosporangium, where they will create new zoospores. And if the animal dies or sheds off its skin, it releases these zoospores again, and the life cycle repeats. Now, it has very specific niches where it lives. So it likes to live in moist environments. So in dry environments, it does not do very well in, and it relies on a certain range of temperature as well. So it tends to prefer colder areas. So it can't survive more than 30 degrees, I believe. And so it's mainly found in amphibians where it's constantly moist. So in adults, it's in the ventral side, the underlayer of their skin. And in tadpoles, it's only observed in the mouth, because that's the only area that has keratin on them. The rest of their body that hasn't developed the keratin layer yet. And so you can see here with a typical healthy amphibian, the skin will be nice and smooth, right? And you can see the epidermal layer on here, just a few layers thickness, and then the dermal layer underneath, and then the tissue layer after that. With an infected animal, you can see that the skin layer itself is completely damaged, disrupted, and you can actually see the zoospores or zoosporangiums themselves on the top layer of the skin. What does the pathogen actually do once it attaches itself onto the skin? Well, here's an example image of a healthy green tree frog and an infected green tree frog on the right side that has been infected with BD. And they're roughly around the same size, but you can clearly see that the infected frog has both redder skin and it's a lot more skinnier compared to the healthy frog. And there's a number of commonalities associated with BD infection. So in terms of behavioral, infected frogs tend to be more lethargic, so they have decreased appetite and they don't move around as often. And they reduce their skin shedding as well, which I'll explain why this is an important factor for the immune system of frogs. Morphology-wise, they tend to have increases in ulcers, hemorrhaging of the skin, and the most common one is reddening of the skin, especially on the ventral side. And instead of shedding off the skin in one whole piece, they will often just shed it in little fragments here, which increases the chances of the zoospores spreading across the environment itself. Physiologically, they tend to have reduced skin function and altered immune function as well. Depending on the species you're looking at, they can have decreased immune function or increase. Another commonality that you often see with infected frogs, especially highly infected frogs, where they have a lot of BD zoospores on them, is low blood sodium and chloride levels. And so these are, they tend to be quite low in their body. And this is the proposed reason why they die from this fungus, is due to heart failure, low circulating ions in their blood. Now, I was interested in how common this phenomenon is in terms of the behavior, physiology, and morphology effects on amphibians. And so what I did was a meta-analysis looking at the effects of BD infection, these experimental studies on BD infection on a whole bunch of different traits. And so I collected 304 different effect sizes from 68 studies, experimental studies, and that resulted in 52 species that have been looked at with these kind of studies. And I've kind of grouped them into 12 categories. So we've got thermoregulation types of traits that were examined, skin integrity, reproduction, osmoregulation, immune function, hormone levels, energy metabolism. So for example, metabolic rate, cardiovascular function. So, you know, heart stroke, heart rate and all that, body condition and behavior. And on the right-hand side are the number of effect sizes that have been extracted for this meta-analysis. Some of you might be aware that Cameron is actually a really great photographer, and he was able to photograph some cool frogs that we had in the lab during my time as a PhD. So you'll probably see sprinkles of Cameron's photos in there now and again. But the important take-home message of this meta-analysis is that a lot of the traits that you see, the responses are quite variable. So for example, with reproduction, there are some studies that show frogs that are infected tend to increase in certain reproductive traits, while others are negatively affected. So what effect size mean is whether there's a positive or negative effect on the animal, but sometimes we don't really know whether an increase or decrease actually means a positive or negative. And so other times we describe it as just an increase in the response relative to an uninfected animal, or decrease in response relative to an unaffected animal. And that's what the positive and negative values mean. And so if it's on 0, it means usually there's no difference or very small change in the response when these animals are infected. The only thing that was consistently affected by that was consistent among species and among different studies was energy metabolism. So their metabolic rate always increased in response to BD infection, which was interesting to see. And there were other traits that tend to decrease on average. So body condition, osmoregulation, behavior, with some slight overlap as well. But on average, they tend to decrease as well. So that's really interesting to see some of the commonalities and differences in responses to BD infection. Now, the reason why BD has such a profound effect on amphibians, and it doesn't seem to be too picky on a particular species, is that amphibian skins are very sensitive to change. And so their skin performs a wide variety of functions. So they use it to breathe, so they can transpire oxygen, they can exchange oxygen and CO2 directly through their skin. And in fact, there are some species of salamanders and frogs out there that don't even have lungs, and they purely rely on cutaneous respiration. Often at times, there's a lot of great studies out there that show that having a healthy microbiome on the skin acts as an innate immune function to protect it against other foreign pathogens as well. So having mutual relationships with symbiotic microbes on the skin is important for these frogs. Now, one that I'm going to pay particular focus on is water and ion regulation. So they regulate their water balance and their salt balance directly through the skin. And frogs don't actually drink water. They just absorb water through their skin, interesting enough. Their skin also acts as an immune defense. So the first line of defense against pathogens, just like with us as well. So they have a whole bunch of antimicrobial peptides on their skin. Another very obvious one is it's just a physical barrier to protect the internal organs against damages, abrasions while the animals are moving, and also, from dehydration, UV radiation damage, and a whole bunch of other chemicals that could potentially impact them. So their skin is important in those respects, and they also use it for acid-base regulation as well. So exchanges of hydrogen ions and carbonates as well to control their pH as well. So really important, whole bunch of physiological functions that the skin does for these frogs. And so any kind of damages or alterations to their skin will impact all of these in some way or another. Now, the reason why amphibians need to regulate their salt balance is because they often transverse in both aquatic and terrestrial environments. And so in freshwater, they have to be hyperosmotic regulators. And it's because they will tend to lose salt through the water through simple diffusion alone if they are not actively regulating their skin. And they will gain water through that as well if they are not regulating it. And on land, it's the opposite effect as well. So on land, they will tend to lose water very rapidly if they have no way of regulating their rate of water loss. And so that will lead quickly to dehydration. Now, some amphibians that are able to live on land quite well. In fact, you guys would probably know quite a lot about the frogs in Northern Territory, especially during the arid seasons. A lot of these frogs, there are actually quite a lot of frogs out there that have excellent ability to prevent evaporative water loss at a very high rate. So, for example, putting sunscreen or this moisturizer on their skin to prevent water from losing out. In this example, tree frog here. So just to give you an idea how ions are important in this context, if an animal doesn't have any way of regulating the salt balance, water will flow from a high concentration area to a low concentration area. So from the inside of the animal to the outside. So in order to maintain skin function, they actually have active transport proteins in their skin that go against the concentration gradient in order to extract the salts in the surrounding water and back into their skin. So these are active transport proteins. Just to give a detail here of how complex this can be, and there's a whole field of osmoregulation or osmoregulatory studies specifically about this. But I just want to point out two key proteins that I'm going to refer to often in here, which are the epithelial sodium channels and the sodium potassium pumps, so ENaC and NKA. Now, ENaC is found on the surface of the skin, or on the, if you're looking at a single cell, it's tend to be on the apical side, so the outer side of the skin. And this is where it's usually exposed to the outer areas, such as fresh water, which has low sodium and chloride concentrations. And so in order to do that, the inside of the cell must have very low sodium inside their cells in order for sodium to passively diffuse in. And within that cell, in order to actively pump sodium against its concentration gradient, the NKA, or the sodium potassium pump, will actively exchange potassium to extract sodium into their body. So this is the active transport mechanism, and that actually uses energy in the form of ATP in order to do this. Okay, so that's how it works in terms of osmoregulatory function. And this will be very important to understand how BD actually affects the skin function from this point of view. I want to add in another complexity because during my PhD, I actually did a preliminary study on cane toads, just looking at the effects of skin shedding on their osmoregulation, right? So it's been known previously that by shedding off your skin, you can actually keep the skin quite healthy and functional. So if for folks that are not familiar with the act of skin shedding or sloughing, it's basically what all these frogs do is that when their skin becomes all worn out and at a regular time interval, they will often shed off their skin in this very predictable behavior pattern by using their forelimbs to push the old skin from their back into their mouth, and they will eat their skin as well. And so what that does is removes the dead skin layer and exposes the new healthy skin layer on top. And what that also does as well, it's because they eat their skin, they also remove potentially harmful pathogens that are on the skin as well, in order to get rid of any potential harmful pathogens. And that has been shown with chytrid fungus as well. By shedding their skin off, they can remove the fungus a lot quicker, and thereby reducing the chances of developing the disease. But what I've also found in my study is that the act of skin shedding also temporarily disrupts their skin function. And this is because when the new skin is fully exposed, it's not completely tightened. And so it's still leaky during this period when they are shedding off the skin. And it takes about a few hours, or about an hour or so, for it to be completely sealed up. And so there's a brief period where they are particularly vulnerable. And if they are infected with chytrid, and especially if chytrid can reinfect them really quickly back onto the skin, then in fact, skin shedding may act as a double-edged sword, whereby trying to remove the fungus as quickly as possible, they may actually further exacerbate the ion disruption or the water and salt disruption that's occurring through their skin in that way. So this is where my PhD study came in, is basically look at how the skin function actually gets disrupted by chytrid fungus. And this is because people have associated linked up skin function with frogs dying from the disease, but providing a mechanisms of how the skin is being disrupted was not really known before this, or not looked at in this level of detail. And also, I wanted to look at the interactions of skin shedding, whether that actually further exacerbates the disruptions caused by chytrid infection. And lastly, I also looked at the energetic cost of skin shedding, because looking at that behavior, that's going to incur some energetic cost as well. And frogs that are typically infected, not just by BD, chytrid fungus, but by all sorts of other viruses and other pathogens as well, they will also increase the rate of skin shedding to remove any potential pathogens. And so that could also incur some sort of energetic cost as well. And so I wanted to look at how much energy they actually lose or actually invest in performing that behavior and rejuvenating the skin. So I took a whole integrative approach. This was done looking at whole animal responses all the way down to gene expressions of those specific proteins that I looked at. And I also contributed to look at some of the comparative approaches at different species as well. And if we do have time, I'll go into that later on. But this was work done with Craig Franklin and Rebecca Cramp at University of Queensland. And most of this work was predominantly done on green tree frogs, and I'll show some examples of other species as well later on. But for the purposes of this, they're all green tree frogs. The first question that I was interested in is, well, how much ions are they losing through the skin when they're infected with BD? And so we did a really simple experiment where we put a frog in a tub of distilled water and a conductivity meter, which measures the amount of salt inside the water. And here's a typical reading of the conductivity without an animal and with a frog inside. And you can see that the conductivity levels are quite low. And so it means the frog is regulating its skin quite well, which is great. We know that the frog is doing its thing. And here's an example of a frog that's in the process of shedding, we call it mid-slough, you can see that the conductivity readings increases dramatically during that time, and it stabilizes there. We can validate this through video observations as well. So we leave the frogs alone, do their own thing, and then watch their behavior through video surveillance. So when we combine this data together with both control animals and non-infected animals and infected animals, we can get this kind of results here. So on the x-axis is the infection intensity. So it's just how infected the frogs are, how much zoospores are on the frogs. And on the y-axis is just the rate of conductivity that's been accounted for with body mass. So this is mass-specific conductivity reading. And so all the control animals or the uninfected animals are in white here. And as you get darker green, the more infected the animals are. As you can see here, when the animals are not shedding, so these are animals that are just, we call them intermolt animals. They're in between their molting phases. So these are just the normal animals here. They increase in their rate of salt loss when they're infected. So this isn't anything new. Other studies have shown this, but it's great that these results are consistent. What was interesting, actually, was that when the animals are shedding their skin, there was actually no difference between an infected frog and a non-infected frog. So the rate of ion loss remains very similar to when there's skin shedding. So what this kind of tells us is that skin shedding doesn't further worsen the rate of salt loss, which was kind of surprising to me. That's kind of counterintuitive to what I predicted, but that's science. So what could explain this? What could explain these results? Well, so if we start looking deeper into what's happening within the tissue level, what we've done afterwards was extract the skin tissue of the animals and put it into this chamber known as an ussing chamber. So it's a chamber with two halves. You put the skin right in the middle, and one has a water side and one has a solution side that is accurate to the internals of a frog. We call that a Ringer's solution. So that's the same salt composition as the internals of frog. And we can see the rate of ions passing through the membrane itself. And we can actually manipulate that by putting pharmacological blockers in there. So one of the pharmacological blockers that I was looking at was the sodium transport system. And so the reason why it's called amiloride sensitive short circuit current is because amiloride is the drug that blocks out the sodium transport system itself on the skin. So you can see here with an animal that is not infected, so an uninfected animal versus an infected animal, and these guys are not shedding. A non-infected animal tends to have pretty high rate of sodium transport, and when the animal's infected, they have a lower rate of sodium transport of extracting salts back into the skin. But the interesting thing is that we found that during skin shedding, there's a huge increase in the amount of active sodium transport being pulled back into the skin. So this could potentially explain why we don't see a difference between an infected and non-infected frog when they're skin shedding. Is because they are actively trying to uptake the salt back into their body, even though their skin might be more leakier when they're infected because of that. And we can show that the skin is more leakier because we can actually look at the skin resistance as well. So the higher the skin resistance, the less likely salts will leak out of the skin. So in this case here, an infected animal that's not shedding will have lower skin resistance. And when the animal is shedding, both when they're shedding, when they're non-infected and when they're infected, they have the lowest skin resistance of all. So if we look even further down at the specific proteins, okay, what's actually happening there? Are they actually producing more of these proteins, or are there certain aspects of the proteins that are being disrupted by the fungus itself? So if you guys remember, one of the proteins that we looked at, that I focused on, was ENaC, the epithelial sodium channels. So we looked at their abundance in the skin and also the expressions of the genes to produce the proteins itself. So if we focus on here, when the animals are not shedding, infected animals tend to have lower ENaC abundance and higher ENaC expression. And what that's saying is these animals are trying to produce more of these epithelial sodium channels in order to counteract the effects of the ion loss that's happening. But something might be happening between that translation of the gene expression to the productions of the proteins that the fungus is doing, that's preventing that. And we can see a similar trend, not as significant. There's a lot more variability when there's skin shedding as well. With the active sodium channel, so the NKA, we see a different type of effect. So with shedding and non-shedding, when they're not shedding, and between infected and non-infected animals, you don't see a difference in their abundance, but you see a huge increase in the sodium channel abundance when they're skin shedding, and that relates back to that increase in active sodium effect as well, with the tissue examples. With the expressions, when they're infected, both skin shedding and not shedding as well, they both increase in their expressions to produce the protein as well. And again, there seems to be some effect that Chytrid's having that's preventing that translation to the protein itself with NKA. And that's only when they're not skin shedding. So that's the proteins that are responsible for actively taking up the salt back into the skin. There's another perspective that we can look at as well. And there was a collaborator of mine, Julia Goldberg, from the University of York in Toronto. She's an expert in looking at tight junctions in the skin that's responsible for the skin resistance. And so she came in with her expertise, and we conducted the study using the same frogs as well, and looked at both the skin function as well in terms of the skin resistance and the protein abundance and the expressions as well of those particular tight junctions. So you can see here with an example, we had a similar setup with the Ussing chamber, where you have the skin on one side, and on two halves we have distilled water and also a ringless solution on the other side. But this time, what she did was put a sugar solution in one of the sides. I believe it was in the water side, and these sugar solutions, they have chemicals in here that are I believe fluorescent, and so if any of them passes through the skin, it can be detected on the other side through spectrometry, and you can actually determine how much of those sugar solutions passes through the membrane itself, so that way. And so what she's found is that as the animals get more infected, they tend to have more of those sugar molecules moving towards the inner membrane. And so they tend to have higher rates of salt loss as well. So again, this perfectly matches what we've observed in our study as well. And one of the tight junctions that she looked at, I'll give you an example here with oculin. So oculin tends to be found in between the cells as well, and that creates the tightness here. So it's typically found between the cells, that's highlighted in red here. And she found a similar set of results where the ocular abundance decreased, but at the same time, their expressions increase as well. So it means the frogs are still trying to make that recovery of their skin function, but seems like Chytrid is affecting those development of the proteins some way. And so the conclusion from the study shows that infected animals tend to have greater continuous salt loss, but sloughing doesn't seem to further increase that salt loss as well. And this is due to the frogs having an active mechanisms to try to regulate their skin function as best as they can during that sloughing process. And we also showed that the chytrid fungus also reduces or inhibits certain regulatory transport proteins, for example, epithelial sodium channels, but not to the same extent as sodium channels as well during sloughing. And also the tight junction proteins seem to also be affected by the fungus itself. So this part provides a mechanistic explanation to why the skin integrity gets disrupted by the fungus itself and how these regulations are disrupted. It also shows that sloughing does provide temporary restoration to the skin function, because we show that the sodium, the NKA, increases in its abundance and the amount of active sodium transport back into its skin. But this is only temporary, because when they are not shedding anymore, that effect dissipates and the frogs have reduced skin function again. So this may actually accelerate disease progression for some susceptible species. Now, if you note here that I've only been focusing on one species at this stage, looking at the really grainy details of how the fungus disrupts these animals. But of course, there are intraspecific variation as well. So some frogs are definitely more susceptible to the fungus, while others are not. And so I also participated in some of the studies that looked at comparative differences in how frogs shed off their skin, and also how they differentially affect the fungal pathogens on their skin. So this is in collaboration with Michelle Omer, who is now in the University of Mississippi in the USA. And she was the predecessor of me. So she initially started off the chytrid work at the Franklin Eco Lab down at University of Queensland, and I kind of followed up on that with a more focus on physiology. But what she was looking at was differences in susceptibility based on how the frogs shed off the pathogen. So you can see here that Michelle looked at 5 different frog species and looked at how different their BD load is, so their pathogen load is, before and after they shed their skin. And what she showed was, for all species, some of them tended to reduce their skin function, reduce their pathogen load on their skin, some more effectively than others. Here's another representation. This is relative change to when they're not shedding. And so for some species, for example, Limnodynastes tasmaniensis, some individuals, there's one individual here, was able to get rid of the fungus completely. So there's a 100% reduction in BD load, and that individual fully recovered. Just fascinating to see. So there are variations to how much of the pathogen load is removed after the animal has shed its skin. You can see here that even within a species, so this example here is with green tree frogs, you can see that certain individuals will continue to increase their pathogen load, while others don't. And the ones that do develop these, the actual Chytridiomycosis disease, we call this the clinical stage, tend to have higher BD load compared to ones that actually recover from there. So this is what it looks like from histological section, where we have an uninfected animal, which is a really nice clear skin. Here's one that is lightly infected, so around 1,600 zoospore equivalents that we counted. And you can see his skin is perfectly fine here. And this animal also looked completely fine as well. So this is just a light pathogen load. When it got to around 20,000 zoospores, you can clearly see the skin function, the skin structure itself completely ruined. And in certain sections, you can actually see the zoosporangium below the skin. So sometimes when they shed off their skin, the zoospores remain, and other times they will get shed off from the pathogen. And so the variations in zoospore load on the skin after they skin shed could depend on their skin thickness and how deeply the zoospores are able to burrow underneath the skin. They don't typically burrow very deep. They only stick to the epidermal layer. So it's not that far deep, but depending on how they shed their skin, this could dramatically change how the infection load changes. So sloughing can act as an immune response. So what I mentioned before, frogs that are infected will increase their rate of skin shedding. And so there are two studies that independently showed us with Michelle's work before I came in, and also my own work as well, green tree frogs. This is one view of it. These are frogs that have different shedding cycles. So we call this intermolt interval, which is basically the time between two molting stages. And you can see here that with a control animal, they don't tend to change much in terms of how often they shed their skin. But with an infected frogs, they shed their skin a lot more. So that intermolt interval shortens. So they shed more often when they are infected. And I also show this as well in my study. As animals get more infected, they tend to further decrease that intermolt interval stage. So they increase in their sloughing rate, essentially. Okay, so I mentioned about metabolic cost of sloughing as well, and also from infection. So we looked at the rate of CO2 production as a proxy for metabolic rate, and this is also mass corrected as well. So we've got non-infected animals in the white here, and when they are not skin shedding, you see a huge increase in rate, the metabolic cost of living. So they have a higher metabolic rate as infection intensity increases. But we saw something similar with the rate of ion loss as well. When they're skin shedding, they have a really high energetic cost of skin shedding, but it doesn't seem to further increase, or not significantly so, when they're infected. If you don't correct for mass, so you just look at the effects of mass here, we've got body mass range, we've got really small juvenile individuals, and rate of CO2 production on here. We've got animals that are, this is just looking at non-sloughing animals. So these are just normal animals that are either non-infected in the white or infected in the green gradient here. So you can see that the rate of CO2 production is a lot higher for infected animals. And with skin shedding as well, there's a further increase. And when they're infected, it seems to be very similar to a non-infected frog as well. But the important thing here I wanted to add is, I didn't mention it in here, the slope also changes as well. So what that represents is that smaller individuals have proportionally higher energetic cost when they're infected relative to a larger animal. So slope is a lot more steeper here because of that reasoning. And so small animals tend to be more vulnerable in terms of energetics when they're infected by the chytrid fungus. So in conclusion, from all of these studies combined, basically sloughing is an overlooked aspect of an animal's immune function and physiology in respect to how animals deal with certain pathogens. And so it's an important aspect to look at, especially for frogs, if you want to look at the interaction between skin function and pathogen effects. So while the sloughing does appear to clear infection in some species, for those that are not able to remove the fungus quick enough, the impacts of the chytrid fungus is a lot greater for them, because they can just reattach themselves back onto the skin much quicker, and that further affects their ability to regulate their skin function itself. On top of that, the increase in skin shedding and the energetic costs associated with that can further increase the energy usage, and that's why a lot of frogs, even while they're still in the active phase, still become more lethargic, and they will lose weight even during the early phases of their infection as well, before they even develop clinical signs of the pathogen. Well, so what are some of the conservation implications of this? Well, there have been various studies that have attempted to look at salt treatments, so providing saline solutions to match similarly to their internal body, to be able to help them regulate their salt balance while the frogs are fighting off the fungus itself. So there's a research group that actually tried to do this in mesocosm settings, and they showed that providing salt into the water bodies around there improved the host survival. So that looked pretty promising. There were some criticisms about doing this on a large scale, because not only are you putting salt into the water, that might affect the whole community in itself. A lot of freshwater animals can't regulate their salt balance very well when they are subjected to rapid change in salinity. So this hasn't really been applied, but it's a good proof of concept that it does improve their survival. And we can also establish a critical period of recovery before the onset of symptomatic disease. So what a previous study found in 2009 is that at a certain threshold, so certain zoospore load, they will quickly degrade themselves to the point where they can't recover back. So what they found is that even providing salt treatments early on during the disease phase, the frogs seem to recover back much quicker. But at a certain point, especially when they're heavily infected, no matter how much of the salt treatments you give them, they don't recover back from that. So there's a certain point where they can't be recovered or they can't be reestablished back, and they die from that. And there's also some should be some careful considerations for smaller and younger frogs as well, because of that increase in energetic and osmoregulatory cost associated with the younger life stage, or just smaller individuals themselves. They tend to be more susceptible at a younger age, or smaller frogs as well, relative to larger frogs. And that's also been observed in the wild as well, where the composition of body sizes seems to shift towards larger ones. So larger ones tend to survive more than smaller individuals here. So that's basically my talk. I'm happy to answer any questions that evolved around that. I know there's a lot happening here, and I'm happy to clarify some of the things that I've talked about. I think this is heavily dealt with in the physiology aspect. So if something doesn't make sense, let me know.
Hi, Nicholas. This is Keith Christian. Can you hear me okay?
Yes, I can hear you.
Yeah.
Nice to meet you.
Well, thanks for that. That was very informative. Really appreciate that. Very, very clear talk. Would you mind going back? You had two different slides related to, you had multiple species and the sloughing. Yeah. So the first, if you go back to the first one, the one before that, I think, yeah. So that I'm just trying to understand the difference between the two slides, because this slide looks like, say for example, Platyplectrum ornatum has a really big before and after effect, but not so much on the next slide. So what's the difference between those two?
I believe this is relative. The second graph looks at relative difference. And so these are, when you look at it on a common scale, the difference is not that great. And compared to here, where they started off with a higher point, and they tend to lose a lot of the pathogen later on. So Michelle did this analysis, and I believe she presented this in the same paper as well. And there were just two different aspects of approach. No, I think these are two different papers.
Yeah,
I have to double check that.
Yeah, that's okay. Now, in that comparison, I would have, or in your work, did you do anything with cane toads? Because my understanding is that cane toads are not very heavily infected with BD in nature. It occurs some, but they don't seem to be badly affected. I would have thought they would make a really good comparison.
Absolutely, that would have been great to do. Unfortunately, I didn't have enough time during my PhD to do this. I did look at the effects of sloughing itself on caintoes. This is kind of starting off early in my PhD, and I needed an example species to work with before I moved on to green tree frogs. But you're totally right. Caintoes have been known to be quite resilient to the chytrid fungus. And other studies have found that it seems to be associated with their skin thickness as well. And the types of habitats that they live in tend to be a lot more diverse and drier in respects that the fungus doesn't seem to grow as well on their skin. So it's that combination of their skin morphology and their habitat that seems to contribute to the lower infection load. And I think they're also more tolerant to it. I can't confirm that, but at least to my knowledge, they're able to resist it because of those features relative to the native frogs. What I do know is that younger cane toads, so especially newly metamorphed ones, they are susceptible to the fungus. I believe there's a preliminary study done, who is it? Laura Branley from the University of Melbourne, and she's done that with cane toad metamorphs, and she found that they were susceptible to the chytrid fungus. So it has something to do with size as well. Cane toads are also larger as adults, so they could hold more of the pathogen and not be affected at the same time.
Yep. Okay, while I've got you, I'll just ask one more question and then I'll let somebody else talk. Early in your talk, you talked about the potential role of the skin microbiome and other aspects of the skin chemistry, the peptides and so forth that are part of the immune system. Mm-hmm. I'm just wondering, just be interested in your take on, I mean, obviously they're parts of the immune system, but there's still plenty of frogs getting chytrid. So, you know, how, I guess the question is how important do you think these different immune mechanisms are, like, for example, the skin microbiome, the peptides, the sloughing, all of those put together is you know, have any gut feeling for which of these is most effective?
Yeah, I tried to keep up with the literature about this, and there's always new studies out there showing, look, we found this particular microbe that seems to be beneficial for this frog, or look, we found this immune function that seems to be helping these frogs to develop resistance. It's very difficult to keep up because there's a whole field in itself just on microbiome, like host microbiome dynamics, and another whole field in itself just on the immune system that I can't even keep up with. But from my understanding, this is going to be very general, is that they all contribute much in some way or another. And this is probably why it is very difficult to predict what species could be vulnerable and what wouldn't be vulnerable to the fungus because of those different interactions as well. Because, for example, skin shedding also removes a whole bunch of other microbes as well, not just the chytrid fungus or pathogens. It removes beneficial microbes. So there is some cost associated with that. But what studies have found, if you live in a very diverse microbiome region, you can reuptake those beneficial microbes back onto the skin much easier than a more monotonic microbiome area, for example. And so all of these features, and of course the immune function as well, will interact with each other in some way to affect their susceptibility. So I don't have a clear picture on this, and in fact, all chytrid researchers kind of know that there's no one-for-all solution for this. So we have to kind of approach it in a holistic sense. But I'm trying to make sure that I'm contributing in some way. You're looking at the sloughing aspect in itself, which tend to be negated in the literature.
Yep. Okay. All right. Well, thanks for that. And thanks again for your talk.
Thanks.
I have a quick question.
Yes.
So your measurements were mostly collected in water. So I'm wondering, how do the impacts of BD or shedding on osmoregulation differ between aquatic and terrestrial frogs? Like if skin is leaky, but the animal's not in water where it can just, something can diffuse.
Yeah.
What's the difference there?
Yeah. So an honor student looked into this with Michelle as well, and they also used green tree frog as a study system. So they looked exactly at that aspect, looking at evaporative water loss, right? So they infected frogs under the same conditions that I was doing my experiments in, but what they did was take the frogs out of the water, put them in a dry environment, and put a fan on them and in a mass balance to look at the rate of water loss that's occurring to the skin, differences between a non-infected animal and an infected animal. And what they found is that their rate of evaporative water loss was a lot higher when they were infected.
What about in terms of like ion transport?
They didn't look into that, unfortunately. So it depends on what species you would like to focus on. Many amphibians will kind of balance out between terrestrial and aquatic environments, but in the end, they will need to go back into the water to rehydrate themselves in some way or another. So that's when they're most vulnerable. If you're looking at an arboreal species, they go less into the water as often, and so their susceptibility would be lower in that sense. But we mainly focus on what happens in the water because that's when they're most susceptible. Because that's also where they get the fungus as well.
Right. Yeah. Thanks. Karen, we can't hear you. Karen? You can also type your question.
So can you hear me now?
Yes, I can hear you.
I just quickly drove over to Keith's place.
Right. Impressive.
Yeah. So my question was about skin shedding and reduction of load, but you also said, they will eat their skin. So I was curious, putting that together, I was curious to know whether or not you can pick up the fungus in their feces and whether or not it's viable.
Yeah, that's a really great question. There quite a few people have actually asked this in the past, like, does the pathogen, so one of the questions that they asked is when they eat the skin, does the pathogen grow inside the body as well? It doesn't, but does it also get expelled out? To my personal knowledge, no, but we do know of another study with ducks where they found the chytrid zoospores inside the fecal matter of ducks, and they proposed that ducks were a method of transporting the fungus to naive ecosystems, for example. So they seem to survive in duck poo, and so it's likely they would survive in frog poo as well. I mean, I've seen a whole bunch of animals survive in frog poo, like blind worms. So blind snakes. So yeah, I wouldn't
So,
be surprised.
Has it been tested?
No, and not to my knowledge, hasn't been tested in frogs.
That'd make a good honours project.
That would be, yeah, and be very simple to do, right?
Yeah. Thanks, Nicholas.
Yep.
Thanks. If there aren't any other questions, we might wrap it up. Well, thanks, Nicholas. Thanks for presenting today
Yeah,
as
it
the
was
last
fun.
presentation of the year.
Woo!
Yeah.
Thank
I'm
you.
sure we'll be in touch.
Yeah, well, enjoy the rest of the year, and hopefully you guys have a very productive new year.
Yeah, you too. See ya.
Bye.